Sasa tukibadilisha mawazo yetu tukipeleka kwenye asexual reproduction. Kwa hiyo hapo tumeona kuhusu sexual reproduction inavyotokea kwenye magrupu yote, Mycorrhizomycotina, Entomophthalomycotina, inavyotokea kwenye Basidiomycota, inavyotokea kwenye Ascomycota. Lakini kwenye asexual reproduction nasema fungi may produce asexual spores by simple haploid nuclear division. Kwa hiyo simple haploid nuclear divisions, kwa hiyo fungi wanaweza kuproduce hizi asexual spores ambao tunapata haploid nuclear divisions. Again, short-lived propagules are produced in enormous number to ensure spread to new habitats. Kwa hiyo kuna tuzalishwa sana zile propagules, kwa kama viuvimbe vile propagules tulivyoangalia kama spores vinazalishwa kwa wingi sana kwa ajili ya kuhakikisha kwamba wanasambazwa na tunawapata wa kutosha kwenye mazingira. In many fungi these are sexual ambao tunaita anamorph au tunaita imperfection au imperfect stage. Kwa hiyo hii asexual reproduction inaitwa kama anamorph au tunaita imperfect stage. Inakuwa na two proved so successful that sexual. Kwa hiyo kwa sexual ambayo tunaita ni teleomorph, ile inaitwa anamorph asexual, lakini sexual reproduction inaitwa teleomorph au tunaita perfect stage, inakuwa na diminished or even disappeared. Kwa hiyo unakuta kwenye hii stages sasa tunasema kitaalamu tunasema in many fungi this asexual reproduction or asexual stage, which is also called the anamorph or imperfect stage, has proved so successful that sexual we call it sexual stage, or we call it teleomorph or perfect stage has diminished or even disappeared. Kwa hiyo hizi species sasa zinakuwa na species za fungi zinakuwa na long been known as a fungi imperfecti, or we call them au tunaita Deuteromycetes au tunaita Deuteromycetes. This by convention contained all asexual relatives of Ascomycota and Basidiomycota, but not those of the former Zygomycota. Kwa sio kama zile za Zygomycota zilivyokuwa. Lakini with the advance in molecular phylogenetic analysis, concept ya fungi imperfecti is becoming increasing redundant as a useful taxonomic group, since most asexual reproducing species can now be placed with their sexual reproducing relatives. Sasa kuna termu ya msingi sana hapa kwa fungi ambayo tunaita conidia. Sasa conidia, tukiangalia wale akina Ascomycota na akina Basidiomycota kama phyla za hawa fungi ambao wanaathiri animals na human at large, the asexual spores, zile asexual spores, zile spores tunaziita conidia. Yani maana yake zile reproductive structure za hawa fungi ambao tunaziita spores. Sasa zile zinazotokea kutokana na asexual reproduction, unasema asexual reproduction tunaziita ni conidia. Kwa hiyo hizi zinazalishwa kutokana na structure ambazo tunaziita conidiogenous cells. Kwa hiyo zile cells ambazo zinaweza kuzalisha conidia tunaziita conidiogenous cells. Sasa hizi conidiogenous cells kwenye some species za conidiogenous cells is not different from the rest of the mycelium. Tofautiani sana na sehemu nyingine za mwili wa fungi ambao tunasema ile hyphae yote. Sasa kuna part kama vile ambazo ziko specialized hyphae ambazo zina develop, hizo inakuwa conidiogenous cells, ambapo conidiogenous cells ni zile cells ambazo zinaweza zikadivide zikatutengenezea conidia. Kwa hiyo hivyo, so tunasema in others conidiogenous cells inakuwa contained within a specialized hyphae structures, au tunaita hiyo specialized hyphae structure tunaita conidiophore, kama mrija ule ambao unashikiza ile specialized structure, unashikiza hizi conidia, hizi conidia. Kwa hiyo kuna conidiophore. Kwa hiyo tunatoka kwenye hyphae inakuja inajiextend au inajispecialize, inatengeneza conidiophore. Conidiophore kwenye apex kule mwishilizo kwake kunakuwa na cell ambazo tunaita conidiogenous cell, ambazo zenyewe zikidivide ndio zinatutengenezea conidia au zinatengenezea hizo spores ambazo zinatokea kwenye asexual process. Kwa hiyo kuna two basic methods za asexual spore production. Jinsi kuzalishwa kwenye zile asexual spores au conidia kuna namna mbili. Namna ya kwanza inaitwa thallic na namna ya pili inaitwa blastic. Sasa thallic, this one is in which an existing hyphae cell is converted into conidia. Kwa ile hyphae tu yenyewe ile zile kamba, ile hyphae yenyewe tu inajidivide inakuja baadaye inatengeneza conidium. Lakini kwenye blastic, this one is a form ni asexual spore productions, ambapo conidium inakuwa produced as a result of some forms of the budding process. Kwa kuna process kama ile budding inayotokea kama vile kwa yeast, basi inatokea hapa tunapata hizi conidia. Tukijaribu kuangalia in details kuhusu thallic conidiogenesis, maana yake kutengenezwa conidia ambazo tunaziita zile thallic. Sasa kwenye thallic conidiogenesis, the conidium is produced from an existing hypho cell. This occurs when a hypho breaks up into sections to form individual cells, au tunaita arthrospores, or when one cell develops a thick wall to form a resting spores, au tunaita chlamydospores. Kwa arthrospores imekuwa derived from the fragmentations of an existing hypha and represent the simplest form of sexual sporulation. In most species of the septum, tunatengeneza septum ambayo ina separate two cells, ina split down the middle, leaving a trace of the resulting torn wall on the end of the spores. Kwa hiyo in a few instances, the arthrospores are intercalated with the separating cells and are liberated after these cells have dissolved. So this leaves a marked annular frill. Ina leave marked annular frill at the end of the detached arthrospores. Lakini kuna aina spores fulani tunaita aleuriospores. Aleuriospores ina represent an intermediate stage between thallic and blastic conidiogenesis. Kwa hiyo kuna thallic ambayo tumeangalia hapa ya conidiogenesis, lakini kuna hii blastic conidiogenesis. So in between or intermediate state inaitwa aleuriospores. These spores are formed from the side or tip of hypha, and during the initial stage before a septa is laid down can resemble short hypha branches. As in all genuine cases of the thallic conidiogenesis, it is not possible for a second spores to be formed at the same point. Kwa hiyo tunaipata sana kwa kina hiyo form ambayo tunaipata kwa kina arthrospores, tunaipata sana kwa kina hawa dermatophytes au fungo ambao wana association na ngozi lakini wana association na plant origin. Kwa hiyo na mimea sana kama kina trichophyton, labda tunaweza tukakutana nao kwa kina trichophyton, wa kina mucorales, na wengine wengi. Lakini pia kuna baadhi ya fungi wa medical importance pia tunaweza tukakutana nao. Lakini aina nyingine hii ya conidiogenesis inaitwa blastic conidiogenesis. Many fungi wana evolve some form of repeated budding that permit them to produce large number of asexual spores from a single conidiogenous cells. Two form of blastic conidiogenesis are not recognized, about tunazijua kuna aina mbili za conidiogenesis ambazo zinatokana na blastic conidiogenesis, ambao kuna ambayo inaitwa holoblastic development, akili kuna ambayo tunaitwa enteroblastic development. Kwa holoblastic, in which both the inner and outer wall of the conidiogenous cell swell out to form the conidium, and we call them enteroblastic development, in which the conidium is produced from within the conidiogenous cells, or the outer layer of the hypha wall being ruptured and an inner layer extending through to become a new spore wall. Or these two forms of blastic conidiogenesis can be further subdivided according to the details of the spore development. Kwa mfano tunavyoangalia kwa mfano in details sasa kuhusu holoblastic conidiogenesis in some fungi, the conidiogenous cells each produce a single holoblastic conidia. In others, however, the first formed conidium produces a second conidium, and the second produces the third, and so on, until a chain of spores is produced with the youngest at its tip. As each conidium can produce more than one bud, a branching chain becomes possible. Example of the mold that produce holoblastic branching chains of spores include species of Cladosporium. In other species, the conidiogenous cells that produce the first formed spores then grows past it to produce a second, which is called the sympodial spore production. That is called the sympodial spore productions. So if this process is repeated, it will result in elongation conidiogenous cells known as geniculate conidiophore. We call them geniculate conidiophore within numerous lateral single spores along its sides. This happens, for example, in species of Alternaria and Bipolaris. Kwa hiyo Alternaria na Bipolaris have some kind of such conidia development. Mode which produce holoblastic conidia we can see maybe in the future presentations. So enteroblastic conidiogenesis in the details in fungi that produce enteroblastic spores. The wall of the conidia is derived from the inner layer of the wall of the conidiogenous cells, and the conidia are produced from an opening in outer wall of the conidiogenous cell. So this permits a succession of the spores to be produced at the same point. The specialized conidiogenous cells from which the conidia are produced is termed as phyloid. Kwa hiyo kunapata phyloid. In some fungi such as species of Aspergillus and Penicillium, continuous replenishment of the inner wall of the tip of the phyloid results into formations of an unbranched chain of connected spores with the youngest at the base, moldy, which produce enteroblastic conidia in chains. Also tutaziona kwa urefu wake labda huko baadaye kama kina Aspergillus na kina Penicillium kwa upana wake. In other fungi kuna baadhi ya fungi such as species of Fusarium and Acremonium. A new inner layer of the wall material is produced from each successive spore. Repeated conidiogenesis results in an accumulation of unused remains of these layers within the tip of the phyloid. The spores are not firmly attached to each other and often move aside to accumulate in a wet mass around the phyloid. Unlike the spores of species of Aspergillus and Penicillium, these spores do not spread on air currents, but are coated with a wettable slime, which appears to be an adaptation to water dispersal. Moldy, which produce enteroblastic conidia, ambao tunaongelea wakina Fusarium, wakina Acremonium, labda tutakuja kuwajadili individually. Kwa hizo ni concept kuhusu hapa, lakini kuna structure ambazo tunaziita annelids. Annelids ni kama structure ambazo tunaziita phyloids. This one are the cell which produce conidia at their tips in unbranched chain, as in the genus Scopulariopsis. Kwenye Scopulariopsis. Kwa hiyo kuna genus hiyo au in the wet masses kama vile kwenye genus tunaita Pseudosporium. Lakini unlike phyloid, kama vile kutofautiana na phyloid, annelids increase in length each time a new spore is produced. So the increase in length each time the new spore is produced, this one are annelids, while phyloid do not do that. All the annelid that has produced many spores will have a number of apical scars and annellations at its tip. Kwa hiyo kule mwisho wake utakuwa na scars za aina mbalimbali ambazo zinatokana na hiyo annellations kama kuproduce zile conidia. So these scars which are left as successive spore breaks off are often difficult to see under the optical microscope. Yeah. Kwa kuna kitu kinaitwa sporangiophores. Sporangiophores tunazipata kwenye Zygomycota in most cases. When the former Zygomycota, one major group, the order Mucorales, often produce the asexual spores or sporangiospores inside a closed sac or sporangium, the wall of which ruptures to liberate them. The sporangium is held above the substratum on an unbranched or branched sporangiophores. The different species of fungi in this group are distinguished from one another by sporangiophores. So sporangia and sporangiophores, as well as by the presence or absence of rhizoids that anchor the sporangiophores to substratum. Kuna stratum fulani kwa kama layers. In addition, these organisms have large accepted or almost accepted hyphae, ambao tunapata aina hyphae ambao haina branching ambao tunaita accepted hyphae. Sasa kuna other forms of sexual and asexual spore productions. Most of them already described, tunajaribu kuzielezea zina identify kwa basics of their asexual reproductive structures and spores, jinsi spores zinavyotokea, lakini ambazo spores. However, there are many. Kuna number of pathogenic molds ambao na produce sexual spores in ascocarps or basidiocarps in culture rather than asexual spores, and these are tutakuja kuziangalia labda kwa kina tunapokuwa tunachambua moja baada ya nyingine. Kwa hiyo hapo tunajaribu kuangalia tunaangalia descriptions, tujaribu kuangalia descriptions of several molds ambao na produce macroscopic fruiting bodies ambao tunaita pycnidia, lakini contain conidia ambayo inaitwa pycnidiospores. In addition, tutaangalia several non-sporing pathogenic molds ambazo zitakuwa pia tutajaribu kuzihusisha pia. Lakini tukiangalia kuna form ambayo tunaita ni yeasts. Yeast are neither a natural nor a formal taxonomic group kama tulivyoona, but a growth group or the growth form found in a wide range of unrelated Ascomycetas and Basidiomycetas fungi. The identification, unlike that of mold, relies on the combinations of the morphological, physiological and biochemical characteristics. Kwa hiyo hizo hapo, kwa hiyo zile characteristics tunazokutana nazo pale. Kuna black yeast, ambao tunaambiwa kwenye science kwamba kuna black yeast they are not of the formal taxonomic group, but the term is applied to a wide range of unrelated Ascomycetas and Basidiomycetas fungi that have darkly pigmented cell wall and are able to produce budding cells at some stage in their life cycle. Many of these fungi form both holoblastic na enteroblastic conidia, sometimes even in some mycelia. Sasa hiyo ni kuhusu concept ya reproduction, jinsi inavyotokea. Uchambuzi wa hizo structure tutakuja kuangalia jinsi inavyotokea kwenye media, kwenye part ya identification ya fungi.